Let me see, get this to work. Are we good? All right. Hi. Yes, I'm Sean. As Dave mentioned, I'm a new postdoc. I'm sticking around with Nancy to wrap some things up. But thanks for coming to my talk, and thanks for the opportunity to speak here. As others have said, it's been a really interesting conference, and you've done a good job of kind of mitigating some of the issues that can happen with a virtual conference. So it's been nice. I'm really excited to tell you, one of my favorite stories about how we can build synthetic microbiomes in honeybees and do kind of something useful with them. So I don't think it'll be hard for me to convince the folks here that there's a lot of potential utility in kind of engineering microbiomes, right? So these microbial communities that we've talked about the past few days are found in lots of like interesting, important places, and they do interesting, important chemistry. So if we could kind of control them or genetically engineer them, there's lots of practical applications that we could do, anything from degrading pollutants or changing carbon fixation or changing nitrogen fixation. There's a lot of work looking at how engineering the human microbiome could be used to improve human health, either by delivering drugs or things like that. But there's still a pretty big gap in terms of what we can talk about doing and what we can actually do. And there's lots of reasons for this. Some of it is that these microbial communities are just really complex, and as folks here know, we don't really understand fundamentally how they're working that well. We lack tools for some of these non-model microbes. Models can be expensive, and it's often hard to translate kind of our toy ideas, into an actual applied use. And so today I'll talk about how we can get around some of these things using my favorite insect, the bee. So yeah, these really incredible kind of gorgeous insects are really important. So I'll say bee through this talk. I'm talking about the Western honeybee, Apis mellifera, and I'm talking about the worker class. These bees, play a really important role in crop pollination to help, provide food. But what I'm really interested in them for is their microbiome. So the bee gut microbiome is pretty unique among insect microbiomes. It's simple, it's specific, it's highly conserved. So there's a core set of taxa that we see in bees sampled throughout the world. This microbiome is socially acquired. The bacteria are really only found in bee guts. Now there are some lactobacillus strains that you can find in other places too. And it's spatially structured. So there are, we can kind of watch this community assemble within bees. And so I'm not going to introduce the whole microbiome, that's a bunch, but the species you need to know about is Snodgrassella alvi. This is bacteria that lives right up against the wall in the ileum and forms a biofilm there. We know this gut microbiome kind of contributes to bee health by promoting weight gain, promoting pathogen resistance, and some immune signaling. But from a synthetic microbiome—
Sorry, I'm having trouble hearing you.
Sorry, that was my watch. From a synthetic microbiome perspective, this system has some real clear advantages. So all of these bacteria can be kind of cultured in the lab. It's pretty easy and cheap to get a lot of germ-free or microbiota-free bees. And so we can actually, hives have thousands of individuals in them, and by going and pulling those bees and raising them in lab, we can get them without their normal gut microbiome. So we can actually get, high replicate experiments where we introduce engineered strains or to find communities into bees. And then as well, the things we learn here, either at a basic level or a more advanced level, have the potential to have actual applied use in a kind of a direct way, right? The model is also something we care about. and bees are under threat. So, you know, this is kind of there's a lot to this topic, and I'll give a very short introduction here, but every year many beehives die over the winter, and those have to be replaced, and that's kind of a costly thing. Some of the major contributors to that are RNA viruses like deformed wing virus, and then these mites called Varroa mites. These mites and the viruses actually act synergistically. So the mites help spread the virus and weaken the immune systems of bees. And so these two things are some of the major threats that, you know, lead to these kind of enormous losses each year now, although there are other contributing factors as well. And there's, you know, a whole field of research that looks at this. So we have a system where it's easy-ish to work with it in lab. We can learn things that we can then directly apply within bees. And, you know, that system is under threat and important. So I think it's a really cool place to kind of explore some of these ideas. And so the story I'm going to tell you today took advantage of a process in bees called RNA-interference. This is a highly conserved eukaryotic immune pathway where double-stranded RNA from the environment is taken up by B cells. Those cells then mount what's called an RNA interference response that leads to targeted cleavage of RNA that shares the sequence. And so this approach, you can actually feed double-stranded RNA to bees and have them mount an immune response like this. It's been shown before to work against deformed wing virus and Varroa mites, but, you know, producing enough double-stranded RNA to feed a whole colony or thousands of colonies is quite hard because it's expensive to produce and it kind of degrades fast. So our plan was to see how we could use microbiome engineering to kind of replace this issue and fix this issue by actually expressing the double-stranded RNA in the bacteria already in the bee gut, right? So you don't have to purify it, you don't have to express a large amount. So this is the kind of plan there. We would take, you know, a construct that expresses double-stranded RNA, put that into bacteria, then give those bacteria to the bees and see if we can induce an effect within bees. So the first part of my work was developing this toolkit. So these bacteria that are found within bees were only isolated 5 years ago now, or 6 or 7. And when they were isolated, there were no genetic tools to work with them. And this is a common problem, not just with these bacteria, but with other bacteria found in, you know, newly isolated communities. So I built this, we call it a bee microbiome toolkit. It's really would be useful in other species as well, but it allows for combinatorial assembly of kind of basic constructs, synthetic constructs that you can then move into, you know, poorly described or recently cultivated bacteria. And it's really based off of this really broad host range RSF1010 plasmid. And so, you know, we showed that we could use this toolkit to engineer bacteria that we isolated from bees, and we used it to engineer beta, proteobacteria, alpha, gamma, and so on. And we used it to engineer strains that we isolated from bumblebees as well. So it's a broadly useful toolkit. But, you know, one of the real questions in engineering microbiomes is, as we take these bacteria and we impose a new fitness cost on them, we impose, you know, the construct of a new device, will they then actually go back in and still function and survive within their host, right? Because we've imposed this cost on them. And so one of the first things we did in bees was kind of put these fluorescent bacteria back into bees. So what this is showing here is just, in blue is Snodgrassella alvi that's been engineered to express GFP. And this is, I think, you know, 5 or 10 days after colonization. So what we can do is, so we can dissect the bee by basically pulling the gut out. And then we can immediately put this underneath a scope, so we don't have to fix it or anything like that. To look and see that these engineered Snodgrassella go back into bees, and they go back to the same place, and they colonize where we would expect, and that's a stable colonization. So then we went in and kind of really wanted to test our idea. So we built this construct here. You'll see it in a few other places, what this is what is on the plasmid. So it's a coding sequence here, and then two inverted promoters that express our RNA. And so this one is directed against a gene called INR1, which is an insulin gene in bees. And we chose this because we could actually look at knockdown of the genes, and then we could look at phenotypic effects as well. So what we did is we took this construct, we put it back into Snodgrassella, we fed that bacteria to bees, and then over the course of 5, 10, and 15 days, we did various tests in bees, including looking for specific gene knockdown as well as phenotypic effects. And so what we show here is that we can actually knock down genes throughout the bee host. So this means within the gut, which is where Snodgrassella lives, within the whole abdomen, and then within the head. So we can specifically knock down this gene. And then we looked for phenotypic effects. So what I'm showing here is that we can actually measure how sensitive bees are to sugar, and we can do this by what's called a proboscis extension assay, where you basically expose them to sugar and they stick their tongue out. And I would include a video because it's quite fun to watch, but I don't think I have time. But yeah, so when we knock this gene down, we see this dramatic increase in sucrose sensitivity, which then when we measure the weight of the bees over 10 and 15 days, bees that have this insulin receptor knocked down actually feed more and they put on more weight over time. So this is just one example of how we can introduce this construct into bacteria, give those bacteria to bees, and see a long-term knockdown. So then we wanted to tackle, the things we really wanted to show, which were, can it be used against RNA viruses and mites? So in this first set, we designed a knockdown construct that targeted a small portion of the deformed wing virus genome. And again, we took that construct, put it into bacteria, fed those bacteria to bees. And then in this assay, we actually injected purified deformed wing virus into bees, and we looked at survival of bees over time. And so what you're showing here is the survival of bees that have been inoculated with bacteria and then exposed to virus. These top three are control conditions, and then this is the actual virus knockdown. We saw improved survival over time. We can do the same thing with mites. This construct is a little bit different. So this construct includes portions of essential mite genes. And what's really interesting here is the double-stranded RNA actually gets into the blood of bees. So when these mites feed, they actually latch onto bees and feed on the bee blood and fat tissue. And so here we're actually activating the mite RNAi response. So what we did is we took these bacteria, we fed them to bees. Once these bees were 5 days old, we then introduced mites, and we let the mites feed, and then we looked for mite survival over time. And again, you can see this kind of dramatic, this time increased death of mites as they were feeding on bees that had this engineered bacteria. So just to review, I hope I've shown you that bees kind of possess this simple microbiome that's important for their health. They can be a useful model for this microbiome engineering, and we can use these engineered symbionts to activate host immunity and actually suppress genes and help them survive viral and mite treatments. But what I'm really excited about is kind of the next steps here, because I mean, I think this is, kind of a cool demonstration of some of what can be done, but there's a lot more that we can build off of this. So I'll talk about that real fast and then I'll be done. Hopefully I'm not too far over. So the first is, actually translating this into an applied tool. There's a lot of work to be done in terms of how we introduce it to hives and how we can actually make a construct that's stable in these bacteria as they're in hives, and how can we measure how these engineered strains move around within hives or potentially move around between hives, and then really looking to see if the health effects that we saw kind of are present in the hive scale too. I think there's interesting biology here. So there's a lot we don't know actually about how the double-stranded RNA gets from the bacteria into the bee, and kind of what process is involved there. So I'm really excited about kind of teasing that apart a bit. And then everything I've shown you has really been, again, just a single strain, but there's a community in the bee gut, and it'll be really interesting to see as we learn more and as we study more how that community affects things like the stability of this engineered device. And then, how can we actually use this community to get a stronger effect within bees, right? Can we engineer the whole community to do kind of the same thing or not? And then I'm also happy to talk about how this might be applied in other systems and what that looks like. So I'll be around and we can chat about that. So that's the end of my talk here. Again, thanks for coming and thanks for watching. I'm on Twitter here. I'm working with Nancy Moran now. The work I showed was with Nancy and Jeff. Luke and Richard were really essential in doing some of this work. Richard will be starting as a grad student at Caltech in the fall, so he's great. And I'm looking kind of for postdoc work starting in 2021, so we can definitely chat about that too. Okay.
Awesome. Thank you much. So we have a little bit of time. And so I just wanted to ask one question to begin with. So in terms of RNA, I am a bacteriologist. Like I have no clue as to how far. So how far does this reach in terms of manipulating microbiomes across eukaryotes, I guess?
Yeah, so it's generally been seen that many insects possess a pathway for taking double-stranded RNA up from the environment. So our RNAi itself as a pathway is present in almost all eukaryotes, but not all eukaryotes are actually sensitive to double-stranded RNA from the environment. Many insects are, worms are. We really don't know what the breadth will be. I think there was some early work showing that mammals generally are not. But I don't know how extensively that's been studied recently or not. But I do think